Just right here. So welcome back. So we're going to continue with the next chapter involving photosynthesis. You are still in the realm of photosynthesis. Okay. Too bad. The nightmare has not ended. Just a quick survey. How did you take any biochemistry class? No. Did you take any biochemistry class? The reason I'm asking this is because this second part of photosynthesis, it involves lots of chemical reaction, tons of chemical reaction. If you look at the light reaction, that's kind of like physics and biology. You know, you're involving with the membrane, you're involving with the chemiosmosis, proton multi-force gradient, and so on. Those things, not so much happening in the second part. The second part, it's more about utilizing all the products from the first reaction. Did you take any biochem? I need to know so that I don't go crazy about it. No? Yes? Okay. If I say non-covalent binding, what do you understand by that? What do you understand by that? Covalent binding versus non-covalent binding. What do you understand by that? What? Okay. Who's sharing electron? Covalent. Okay, that's good. That's good. What about if I say this thing? Have you seen that word before? Adjunct? No, not adjunct professor. Although kind of similar actions happening. That's kind of like sharing electron. You see, if you have let's see. Cannot believe I'm teaching this now. And this is what molecule is this? So they've got 2 carbons, so that's what will be ethane. Ethane. Let's see, ethane can react with what? Give me one reactive thing. Bromine. Br. What you gonna get? So when you have, this is bromine. When you have the first molecule react with the second molecule, you know that there's going to be some kind of electron sharing, right? Right. So what are you going to get when this thing to combine together? So you're going to get something like this. Remember, okay? C, that's the minimum occupation of carbon surrounding it. So now this double bond is removed. You're going to get bromine down here. H. So what's the name now? So we got 2 bromine. Hence the word is bromo ethane. This is adjunct on top of each other. So familiar with this up to this point? Okay, that's good. That's good. What about if I go crazy with words like this? How about carbamylation? Have you seen this before? This is actually just kind of some kind of chemical group, like this thing. Added, you got a fundamental molecule. This molecule, something decide to attach something else to it. So these are all the, what do you call that, functional group. Have you seen this before? No? Just esterification. Isomer, okay. Esterification, people do to do what? Perfume, soap, right? Isomer? What is it? What does it show? Actually, from the word, actually you can tell. So if I talk about glucose, how many isomer there will be? Possible. Isomer is actually to show that it could be like many form of it, but actually it's the same molecule. Like the L, like the D glucose, L. So are you familiar with that thing? No? Yes? No, because you like glucose now, we are talking about the same molecule, but slight arrangement of this carbon or functional group, one molecule go up, the other molecule is the mirror of it, will change the conformation of this isomer. Did you learn that? No? No? Yes? Oops. Why this thing? I think this is already whiteboard. Keeps doing this. So macam mana nak tulis lagi? That's not it. That's not it. That's not it. Anyway. Okay. Not to worry too much about it. You in the notes, you see all the crazy notes here. You're going to see these biochemical words more and more again. It is not expected for you to be able to draw the chemical molecules or anything, but you understand the existence of such a thing. Okay, carbamylation. So these are all events in biochemistry. When this event takes place, it has impact on other molecules. When this happens, different thing will start to happen. Originally, that's not happening, like the formation of soap. Right? The formation, the latex. You know, latex people used to make the aeroplane tires. Latex, in the form of what? Was it solid or liquid? Latex. Liquid. Aeroplane tires? Do you think the tire is super strong? The latex liquid, is it strong? How come it becomes strong now? Yeah. People put in what? Yeah. So that's vulcanization. So things like that. These biochemical events happen. It changes the properties of the substance. Now the substance becomes something else that has a completely new properties. It could be better, or it could go the other direction. Okay, the same thing is happening in photosynthesis, right? So when all these molecules are being subjected to various chemical reaction, they start to change form to become a product of different molecules. Maybe the chemical formulas, the number of carbon may be still the same, the number of hydrogen is still the same, but these slight changes will change the game altogether. Okay, so bear that in mind. So what's this about? So this is about the second part of the photosynthesis, which is the carburation. Why is it there's no that thing is not on. Okay. The biochemical reaction in photosynthesis is the very process that give life to this planet. Okay, because the photosynthesizing organisms, not only plants, you have learned now that some bacteria can do that, algae can do that. These photosynthetic organisms, they have the capability to reassemble molecules. In the case here, it's carbon. Assemble carbon. Carbon is it's everywhere. Okay, but if you're talking about, give me a product of carbon, for example, diamond. Can you eat diamond? Will that satisfy your energy needs? Just use diamond as your popcorn snack. It's not still carbon, but that's not the kind of form which is going to be useful in terms of energy expenditure needs for the organism. So this second part of photosynthesis, namely the carbon reaction, got so many names, okay? The carbon reaction, the Calvin-Benson cycle, the pentose phosphate pathway. There are so many other names. These are all synonym, okay? This particular process get energized and then reassemble this carbon in the form of precursor to sugar. Okay, so that's the basis of it, right? So in order to do this, let's appreciate something about the amount of energy that comes to the earth. Okay, all our earthly energy originates from the suns. Okay, so that's the amount of energy that comes to the earth. 3 times 10 to the power of 21 joules. I put it down here to give you some kind of appreciation what joules is like, because you deal with this on a daily basis. Okay, from this amount of sun energy that comes, about half of it is turned into carbon. Okay, what about the rest? Or some are lost, back, got reflected to the outer space and so on, because nature's biochemistry and biological processes are not perfect. Okay, there's bound to be some energy lost somewhere. But remember, like what Einstein said, energy cannot be created nor destroyed. However, it can be changed from one form to another, or recycled from one system to other system, right? So this sunlight energy turns into this carbon, and guess what? This amount of carbon that get fixed per year is the one that gives this planet the identity, gives our humankind the history of our anthropological history. Without this capability, by this carbon reaction, would you have your ancestors? Your ancestors needed to eat as well. Pharaoh needs to eat as well. All the bad guys and the good guys need to eat. Everybody needs to eat. And not to mention that, all the animal, all the microorganism, everybody needs to eat. Okay. However, these are not possible if the very process that turned sunlight energy into carbon form of concentrated energy is possible, right? So take that into perspective, right? So these are the foods that we're talking about, and guess what? These are actually originated from plants, right? You got your carbohydrate, that's the amount of joules. remember, okay, joules and calories, these are interchangeable. You can multiply calories into this 4.184. Some food molecules are more energy dense compared to the others. In our case here, which is the most energy dense? Fat. Gram by gram, fat contains more energy than the other macromolecules, nutritional macromolecules, okay? And another concept is enthalpy and entropy. So in order to make all of these molecules possible, it needs to be built up. Entropy needs to be prevented from happening. It's like you are building a tower. So long the tower has not finished, do not let any bombs come near it. Otherwise, you're going to crumple down and you're not going to get your tower. Okay, that crumpling down of a structure, that is entropy. Entropy usually happens when there's some kind of disorder of molecule or atomic disorganization, right? And talking about the photosynthesis, one thing you need to understand, plant is not the only one that can do this carbon reaction. Okay, if you remember this diagram here, when you learn about the endosymbiosis, which you got two ways, the primary and secondary endosymbiosis, this actually gave rise to the common ancestor for all photosynthetic organisms on this planet, especially the first round here. This organism here, Archaeplastidae. I think there's a word somewhere in here. Ah, yeah, this one. Archaic, it means ancient, very, very old time ago, right? So this common ancestor undergoes evolution, million upon million upon million of years, and then from these three lineages of photosynthetic organism came about. So we got the Chloroplastidae line, and then we got the Rhodophyceae line, and also the Glaucophytae line. So Chloroplastidae includes all the green plant and the algae, the green algae. Anything to do with algae, under this line. Okay, Viride is the Latin, it means green. Right, and then you got Rhodophyceae. Rhodo means rose, that's Latin as well, hence the red algae. And Glaucophytae. Glauco means, what does it mean? Grey. It's a Latin word as well. It means grey. So these things, they further develop, further undergo speciation, adaptation, acclimatation until to the present day, right? So the plants that we're dealing with now today in this region, and then you got your seaweed. Guess what? Do you know any organism that goes into this red region? Your agar-agar. You know, you go syrup, agar-agar syrup though. That agar-agar, if you go to the supermarket, the one that looks in the strips though, belongs to this region, right? And then you got your cyanobacteria here, like spirulina, right? So it's a long process to come about until that we are here today. In fact, there's a small interesting fact that all the carbon that get assimilated on this earth, resulting to the oxygen that we breathe in, actually not very much coming from the terrestrial or land plants. It's actually coming from the other two here. Because these two in the sea, on earth, sea is more or land is more? You got more sea and more ocean. So these two contribute so much of the oxygen that we consume. So very, very much. Okay, right. Right. Okay. Now we have a quick recap. So we have learned the first part of the photosynthesis, still in the chloroplast, but different regions in the chloroplast. So we learned about the thylakoid membrane, and all the associated light reactions that happening to it. To get products of light reaction, what are the products of light reaction? ATP. What is the function of ATP? To give energy, to phosphorylate anything, because the P can be donated to somebody else, and the process is called phosphorylation. And then you got NADPH. Stands for what? How do you say this in words? NADPH. Tak ada. When you say that first word, that itu dah salah sebab ada H kat depan ni, ada ujung ni. Reduced nicotinamide adenine dinucleotide phosphate. Kalau NADP+, you can say nicotinamide first. However, you got H here, reduced. Reduced is very important, meaning that it has extra electron that it can donate. Very important, right? And then we got a byproduct as well in the form of oxygen, right? We're not going to be talking so much about oxygen. We're going to talk about that later when you learn about respiration. Now we're going to focus on the other two products, the NADPH and ATP. So ATP is the energizing phosphorylating molecules, and NADPH is the reducing molecules. These two are needed in the second part, which is the Calvin-Benson cycle. Actually, there's three guys, which name should be here. Calvin-Benson Bassam cycle. I think they dislike the Bassam a bit, so they keep the Calvin-Benson only. Yeah. This is it's a unicellular cyanobacteria, which do photosynthesis, just like plant. But you can see that the first requirement to do carbon reaction is you need the carbon. Okay. One thing about carbon is it's not really, you can say that it's not very soluble, CO2. Okay. It's not like sugar. You just put sugar, it goes away. It becomes part of the water now. So CO2, it is soluble in liquid. Okay. But there are many things that can determine its solubility level in the liquid. For example, temperature, and also the pH level, right? So when you need carbon for carbon reaction, while the carbon itself has some kind of solubility issue, a special mechanism is needed to trap this carbon. Okay, so in other photosynthetic organisms, you will see they have specialized structure to trap this carbon. Okay, and this trapping of carbon is collectively known. You can read more in this web topic 10.1. I'll put later in the folder. CCM, carbon concentrating mechanisms. Okay, unfortunately for plants do not have special structure in the chloroplast itself, to concentrate this. But in bacteria, we got this. So we got two organisms here. We got the Cyanobacteria on the left and Chlamydomonas on the right. So in the Cyanobacteria, the CCM structure is called carboxysm. I think there's a word in here. Yes, this word. Carboxysm. And this is the structure here. For Cyanobacteria. For Chlamydomonas, which is an algae, remember, this is the glycophytae, one group of photosynthetic organism. This belongs to the algae. Okay. For the Chlamydomonas, they do not have the carboxysm, but it has something else. This thing, P, pyrenoid. What's this pyrenoid? Pyrenoid. Okay, so depending on the photosynthetic organism, they have different strategies to concentrate the carbon. Okay, so wait, what about the plants? So it's a bit unfortunate for plants. Plants do not have this specialized structure in the cell or in the chloroplast itself to concentrate this carbon. Okay, so meaning to say, in evolutionarily speaking, the plant has not fully evolved yet. Otherwise, they would have this special structure. You know, there are some labs that I know in Australia, they are trying to introduce this pyrenoid into plant. Because pyrenoid, for some reason, kind of work well with chloroplast. Right. If they got that, that would be a huge leap in scientific discovery. Okay. Right. So let's look at what happens actually in plant, even though it do not have the CCM. So how does it achieve the carbon cycle? First thing first, the carbon cycle or the Calvin cycle has 3 phases. Bear that in mind. These 3 phases are carboxylation, or the other name is carbon fixation, followed by reduction, meaning that you change the molecule of the cycle by allowing it to receive extra electron, and then regenerate again, the whole thing, right? So if you look at in the terms of the cycle, it looks something like this, okay? Very easy, 3 phases. However, the detailed story for each of these, that's what I'm gonna make people cry. Yeah. So let's read this. Carboxylation, it's actually the action of the existing sugar molecule. You see, in the chloroplast, there's already a 5-carbon sugar molecule, which is called ribulose 1,5-bisphosphate. 1,5 meaning that in the carbon change, carbon number 1, carbon number 5 has phosphate group attached to it. Okay, just to refresh your biochemistry, right? And this sugar molecule come together with CO2, not in the form of gas, but in the form of liquid, okay? Liquid CO2 fuse together by the power vested by an enzyme, catalytic enzyme called Rubisco, okay? Right, so that is the carboxylation. And then when it enters, this will give rise to a molecule, 3-phosphoglycerate. I should warn you, because you're going to see a lot of this a lot later. Due to this biochemical reaction that can act upon one molecule, esterification, carbamylation, isomerism, you're going to see so many things that is actually the same thing, originally the same thing. So it starts with glycerol, glycerate, glyosylate, I think, and so on. There are so many. So these are all start as glycerol. There's another one, glycolic acid. You should know glycolic acid if you go to Watson all the time, right? There's a lot of that on the cosmetic shelf. What's that for? Glycolic acid. Or you don't go to Watson. So these things, due to this chemical reaction, we're going to see later, it will change the form from glycerol to glycerate, glyosylate, and also the glycolic acid. Okay, but they are actually basically glycol thing, right? Right. So in here, it is phosphoglycerate. It is actually glycerate with a phosphate attached to it. Then these products enter the reduction with the help of ATP and NADPH. This is further changed into another new molecule called glyceraldehyde 3-phosphate. You see, the 3-phosphate is not changing. It's just it becomes a new thing due to the reduction action acted upon it, right? This is very critical here. You see, there's an opening here, meaning that the product at the end of reduction phase, some exits, the rest continue. Very important. Not everybody exit. It doesn't work like that because this is cycle, right? So the one that exits becomes the sugar precursor that I was talking about. Why do I say sugar precursor? You learn so far, it's all about glucose, right? Photosynthesis produces glucose, photosynthesis produces glucose. That is actually incorrect. Whether it gets turned into glucose or not, it's plant's decision. But what's for sure is at the cytosol level, cytoplasm level, two things get turned into first. It gets turned into starch and also sucrose. So this sugar precursor in the chloroplast, correct? Then it will exit chloroplast. Where is it now? In the cytosol. So in the cytosol, it will be changed into starch because that is the storage form of it. Yeah. However, not all of these can be turned into a storage form. Some needs to be transported into the baby leaves. So if the plant decides some of the sugar needs to be transported away, it is not going to be changed into starch, but changed into sucrose. So sucrose is the transport molecule. Okay, why sucrose? You see, did I mention anything about glucose here? Actually, plant decide to do the glucose if it needs the further ATP because glucose never leaves the cell. Remember, glucose that has been formed in the cell never leaves the cell itself. The only thing that leaves the cell is actually sucrose. Why is that? Glucose is very reactive. It's a reactive sugar, meaning that it will cause havoc with whatever it touches. That's why you cannot have so much sugar in your blood. Okay, it's detrimental because it's very reactive. So the sugar, if it turns into glucose, it will be quickly transported into mitochondria for cellular respiration. You see, very brief actually, the lifespan of a glucose in the cell, plant cell. It gets manufactured, the glucose, then straight go to the mitochondria to be broken down so that more ATP is produced. Do you see anywhere in here glucose gets stored? No. No. So that's why it is a struggle with us human because we always have some kind of glucose in our blood. We need that, but not too much. Okay? Right. So that's the essence of this. And what about the remaining of it? The remaining of it, of this end product, G3P, usually people call it G3P, will proceed to the beginning in order to produce this again, ribulose 1,5-bisphosphate or RUBP, so that the cycle does not stop. It will continue on, going on and on. Don't think there's only one cycle in the chloroplast. In the chloroplast, in the stroma of chloroplast, there are millions of these going on at the same time. Okay, so they kind of complement each other at different speed. So if you look at, let's say that at 10 a.m., at any given time, some Calvin cycle at this stage, some 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 at this stage. So all the stages are present at any time because they cannot stop to happen. If it stops to happen, it will cause backlog. It will stop the sugar production. Right? Talking about probability now. Right. Ooh, what's that? Okay, relax, relax. These three phases of Calvin cycle, you have two ways to look at it. If you want to look at it in the form of chemical reactions, what chemical components reacting with what, you go with the left one. Okay, the carboxylation phase, reduction phase, and then regeneration phase. So you can see the complexity of it, of this molecule. The good thing is you can already see, oh, this is the stage that uses ATP and NADPH, and then this thing is actually, they kind of split before they made up at the end, right? So you get the appreciation of it. Another way to look at it is just look at the summary. This, when you are interested with the reactors, the products, and also the enzymes responsible for it. The reason we need to have this, not only this, even though biochemistry students kind of, very, very, very fond of this diagram. The reason we are dealing this because you learn physiology. When you learn physiology, you understand that in any biochemical reaction, there is always a limiting factor. You need to ID the limiting factor so that you understand how the plants speed up the reaction or slow down the reaction. Because at the end of the day, it's the very process that is manufacturing your harvest. Right. Right. So look at this. The number one, the one that I highlighted here, is actually belonging to the carboxylation phase. So what is the reaction here? RUBP plus CO2 plus water. Now you learn something new. Maybe before this you have learned Calvin cycle, but you didn't know Calvin cycle need water as well. Yes. Calvin cycle, just like the light reaction, requires water to run. In the light reaction at the thallic membrane, the photolysis, you use the water, split the water, so that you get what out of the water? Electron. Yeah, you get the electron. So in this process, you also need water, but not so related for the electron transport. You need the water because to hydrate the molecule, right? Sorry. Right. So the product of this is you get 2 molecules of phosphoglycerate. Actually, it was originally 1 molecule, but this molecule, scientists omit this because it's very unstable. Scientists go straight to 3-carbon phosphoglycerate. How come? Why 2? RUBP, 5 carbon. CO2, 1 carbon. You bring together, you will get 6 carbon molecule. These 6 carbon molecules, super unstable, maybe like less than a second, it will quickly broken down. It breaks down, it becomes 3 phosphoglycerate, 2 molecules of it, right? So what actually happens here in this first phase of Calvin cycle will greatly determine the activation of the Calvin cycle, will determine whether the Calvin cycle starts or not, will determine the rest of the steps here. You see, there's so many steps in here. 12. Actually, there's more if I use other book. Then you will cry even harder, right? Right. So let's look at what happens actually a bit detail in number 1. In here, in number 1 here. So in number 1 here, this is what happens to the RUBP the 5-carbon sugar, and also the Rubisco. So this RUBP, it will go some kind of chemical transformation to itself, right? Isomerization and condensation, because this RUBP, depending on Rubisco, the enzyme that acts upon it, will either bind to oxygen or CO2. And that is the problem with Rubisco. That's not you cannot blame RUBP, okay? Blame Rubisco. Rubisco. So the problem with Rubisco is it has carboxylase reaction, and also it has oxygenase reaction. If the Rubisco take CO2 and then fuse with RUBP, you go up here. You will get the one that you saw earlier, the 3-phosphoglycerate. And then this will proceed to photosynthesis, 2 molecules of it. However, if this Rubisco decides to do the oxygenase, it gets the oxygen, the liquid oxygen, fuse with RUBP, then you go down here. It will produce 2 molecules as well, but only one is phosphoglycerate. The other one is phosphoglycolate. Yeah, these 2 phosphoglycolate has got this synonym. We call it the C2 photosynthesis. All this why you learn about the C3, C4, and CAN photosynthesis, right? That's another one, C2 photosynthesis. Who is the C2 photosynthesis? This thing, photorespiration. So photorespiration is actually the C2 photosynthesis. Is it really photosynthesis? No, it's actually carbon salvage pathway, right? So what seems to be the issue here? If it proceed with this, you get your sugar. But if it proceed with this, you only get half of the sugar precursor. Not only that, the moment phosphoglycolate, plus another one, glycolate. The moment this thing is manufactured, it's actually a bit toxic. The plants need to deal with it. When the plants need to deal with it, not only that it will use energy, it will cause other organelles to intervene as well. Okay, this glycolate needs to be detoxified. To detox glycolate, you need 3 organelles. You need the chloroplast, you need the peroxisomes, you need the mitochondria. We will learn, I think next week, about this photorespiration in detail. But for now, it's good to know this thing can happen. When does this thing happen? When CO2 concentration surrounding Rubisco decrease. When it decreases, when the temperature is high, when it gets too acidic. When else? This is why plants are not doing well in the field when it's too acidic. This is another one reason. Usually you learn because of the nutrient availability, right? When it's too acidic, some nutrient is not well. Well, that is one thing. That's another thing. You create acidic environment in the chloroplast as well. When it's too acidic, CO2 cannot attach to Rubisco properly because it's competing. That's the keyword. CO2 competing with oxygen for the catalytic site of Rubisco. As simple as that, right? Okay, let's see what happens. I'll try to finish in 10 minutes, right? So now, this product, phosphoglycerate, will enter the second phase of the Calvin cycle, which is the reduction phase. Reduction has nothing to do with something get trimmed off, something get pruned off. We are talking about biochemistry, meaning that it receives hydrogen. As simple as that. Reduction. Reduction in chemistry means somebody is receiving hydrogen. Right. So what happens here? So I have highlighted here. These 3-phosphoglycerate will react with one molecule of ATP to become 1,3-bisphosphoglycerate. And this process is what we call as phosphorylation. So the phosphorylation of 3-phosphoglycerate, this phosphoglycerate, sometimes people would put it PGA, phosphoglycerate. It depends on the word. That's why I'm not so big on the this abbreviation. You read 10 books, 10 books will have different acronym abbreviation for it. So for now, we use the full name. Right. ATP is added to phosphoglycerate. Now, carbon number 1 also has phosphate. 1, 3. Carbon 1 got phosphate. Carbon 3 got phosphate. But it is still glycerate. It is still glycerate. It's not changing to anything else. Okay? Right. This 1,3-bisphosphoglycerate will then receive hydrogen. Will get reduced by the action of NADPH. Now it becomes a new molecule. You see, it adds the name here. Glyceraldehyde 3-phosphate. Yeah. In this process, you can see that there is no more 1,3. Carbon number 1 get dephosphorylated actually. That's why at the end of here, you get NADP plus inorganic phosphate. A phosphate group is released from the 1,3-bisphosphoglycerate. Why it has to be removed? If it is not removed, how can it receive extra hydrogen? Right. That get removed. Now it becomes the new G3P. Some books say it. It's good to know a bit. This thing cannot. Glyceraldehyde 3-phosphate. And guess what? This is the sugar precursor. 2 times glyceraldehyde 3-phosphate, you will get a glucose. But there's no glucose happening right now. No. Remember, who decides to get glucose, sucrose, or starch? The plant will decide. Depending on the energy demand. Depending on whether it has babies or not. Maybe some plants, for example, like the perennial plants, it will decide during the fruit season to shuttle all the sugar to the fruits. But during non-fruit season, it only has what? Less organized, only leaves, no fruit, right? So it will decide much of it go to starch, only some leave as sucrose to the leaf. Because leaf is not really a syn sugar. It is a syn organ. But if you compare leaf to fruit, which is the stronger syn organ? Fruit, right? Is that? But some leaves are sweet, right? What are example? Sweet leaf. Cannot think of one. Lalang. Cukup manis. You know that plant? Tak tahu. Pokok asin-asin. Sauropus androgynous. Sauropus androgynous. Cukup manis. That leaf is sweet. Okay. Right. So, up to this point, you can see that ADP and ATP has been used. Right? Right. So, whatever that I've said just now, that is the summary of it. To get this. Remember, at this point, the Calvin cycle not only fix one carbon with one RUBP at a time. No, that's not what's happening. Okay. actually in reality is in order, because one need to exit. If you start as one carbon and one RUBP, when it reaches at this point, everything needs to exit. Who's going to continue? Right, you cannot continue. So the reality is actually 3 times RUBP react with 3 times CO2. So the total carbon here is 18. 18. When it reaches here, it reaches in the form of 6 molecule of glyceroldehyde 3-phosphate. This is 3 carbon, right? If you times again, the carbon is still 18. 18 carbon. Remember, one need to exit here. So when it exit, now it becomes what? Whose exit, who continue? Exit is 1 times G3P, which is 3 carbon. Whose continue? Regenerate. 5 molecules of G3P, which is 15 carbon. Okay, this is how it works. And these 5 molecule of G3P, which you will, I think we will just stop up to this point, it will continue to become only 3 molecule of RUBP. It becomes this way. Oh, see, you start with 3 again. Can you see the magic here? It starts as 3 RUBP, react with 3 carbon. The total is 18 carbon. At the end of the reduction, it is left with 6 molecules of G3P with the total of 18 carbon, right? However, not all 6 molecules of G3P will exit and do sugar, do starch, do whatever. Only one precursor, hence the word precursor. That's why it's called C3 photosynthesis. This is why, because the first product is G3P 3 carbon, glyceroldehyde 3-phosphate. 1 carbon exit. So what about the balance? The balance is 5 units of G3P proceeds to regenerate. During regeneration, a shuffling is involved, like you are playing a Lego and cut. Reshuffling is involved. Originally, you start as 5 molecules, then it reshuffle, boom, you come with 3 molecules of RUBP. This is 5 carbon, right? What about 3 molecule RUBP? How many carbon? Still 15. And you get exactly the same. You end with 5 carbon, you start with 5 carbon as well. 15 carbon. 15 carbon. This is 15 carbon. So that is the reason why this has to be in the form of a cycle. It has to be sustainable. In any way, RUBP level, because plants do not have time to make new RUBP, it needs to find a way. It's like a riddle. How can we use this sugar without losing the sugar at the end? That's the riddle. So evolution, all the magic happens millions of years. They come up with this strategy. Very smart. Who tells the plant to do this? That's a mighty good question, right? Who tells the plant to do this? I mean, like even in the lab, you will find hard time, okay, to ensure that your reactant stay the same number at the end. The plant can do that. So it just use this carbon dioxide. So when this happens, the regeneration is taking place. RUBP is not sacrificed in any way, because in order to make RUBP, energy is also needed. If the plant is busy making RUBP, the sugar, the 5-carbon sugar, when does it have time to do all of these complicated things? It just do not have time. So some said maybe, I think, maybe this is not right. They say that RUBP is actually made at night. Right, because nobody need it. You know what? We just make the curry puff at 4 a.m. because lapar nanti nak jual. At 4 a.m. nobody wants the curry puff because nobody is awakened. Right. But the moment they have awakened, jual lah. Right. Okay. So up to that point. Right. 5 more minutes. So if you proceed with the regeneration, you will undergo, tengok, ada 10 steps. Matilah. Habislah. That looks very worrying. So what happens to these 5 molecules of G3P? So these 5 molecules, like I said, they will undergo reshuffling, rearrangement within these 10 steps of catalytically active reactions. You can see that there's so much enzymes being used here, right? It involves isomerization, it involves phosphorylation, and some of these actually use ATP here. Can you see anybody using NADPH? You see? Now you learn another thing. There is an imbalance need of ATP and NADPH. You see, NADPH kind of used once here. However, ATP, you need here. When you want to regenerate, you want ATP again. So that's why when you learn the light reaction, plants decide to do cyclic photophosphorylation so that more ATP is generated to satisfy the needs of ATP in the Calvin cycle. Right? So, in order to get 3 RUBP plus 3 carbon, how much NADP that you need? How much? Actually, for 1 CO2, right? Sometimes I forget this as well because my brain, when it comes to number, just refuses to remember. 1 CO2, it will need 2 NADPH plus 3 NADPH. Is it written in there somewhere? I think it's written there somewhere. Oh, yeah, I put it here in summary. This sentence here. So at the end of the cycle, in order to assimilate each molecule of CO2, you will need 2 molecules of NADPH, 2 molecules of NADPH, and 3 molecules of ATP. Sorry, terbalik. Lupa. ATP. Apa NADPH 2 kali? ATP. Yep. But that is only 1 CO2. If you are talking about the net of it, this is the net, okay? Net. This is called net energy use. Because only this is the only one that will exit to get your sugar precursor. But the reality is you use 3 molecules, right? If you are following the reality, how much actually NADPH and 3 ATP? Darab je lah. So, ini 2 times 3, 6 NADPH. Lepas tu, 3 times 3, 9 ATPs. That is 1 complete cycle. This is the net. This is complete cycle. Okay, right. Okay, so up to that point now. We'll continue this again. This is just how to activate the why light is needed to activate. Even though the Calvin cycle do not need light directly, it need light to activate the enzymes. Okay, right. That's why when it's good to when you talk to farmers to make farmers understand light is also an input for the agriculture. Without light, you're not going to get your crop. Right. Okay, I think that's all for now. We'll continue later. Any question? All good? What week is it now? What week number? 6 ke 7? Kalau 7 bukan kau dapat hantar assignment ke? Kau macam mana? Habislah. Is it 6 or 7? 7. So next week holiday. Next week, but the other day I asked whether 7 or 8 weeks before mid semester. You said 7. Kalau 7, kalau this is already 7, maksudnya next week should be mid semester break lah.
Kata dia orang tapi tak ada delay kan? Tak ada inform dia pun tulis juga kalau jaga orang tak check setiap.
What does academic calendar says?
Dia just tukar je. Tak ada notification ke apa.
So, okay. So, meaning that you still have class next week? No, I need to know because we need the time to go through your assignment. We need to evaluate your understanding about all of this, right? And I'm not going to do it one day. We got 2 days. We can do one. That will be your marks. Kau tak nak markah first test. So next week not yet holiday, is that correct? So maksudnya for this semester betul lah 8 weeks tu. So siapa cakap aku salah hari tu? No, I didn't know. I kind of heard somebody cakap benda tu. That this semester is not 7/7, it's actually 8/6. I kind of heard that.
Itulah sebab kalau kita check first awal semester, kita ikut lah. Tapi apa nak check jadual tu setiap minggu?
They like to change stuff in the middle, right? Which is tak boleh lah macam tu sebenarnya.
Tapi bagi tahu buat keliling ke surat ke.
Maybe they want to for the convocation. Yeah, convocation. The one that going to be available for the Salam Sauk is not available on the 7th week, but available on the 8th week. So they push everybody 1 week. Okay. You know when you go to graduate, you shake hand with that akong, right? That shake, every shake RM30 tau dia dapat. Bukan kau, dia. Kau nak seronok sangat. Tu namanya Salam Sauk. Ting 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 ting. Kan ramai ada pokok-pokok kat dalam assignment. Your glossary will check again next week. I think we can make both on Monday and Thursday. All right. Okay. So quickly for this afternoon, go straight to the farm. Is it raining? I think we better start. It's always rain around 3, around 4, right? 80% means it's going to rain, very sure. Be sharp there by 2. Okay. There is a slight change in the treatment that we're going to give because I look at the plant. I kind of decide to change it a bit, which is good because you got to learn something a bit more. So I will explain later at the farm. So today we're going to apply the foliar. Last week your friend applied foliar as well. Okay, this foliar is not treatment. Okay, this is actually given to all the treatments because you don't apply the granule anymore. Right, you only apply one time at the beginning, and that is slow release. But what about the rest of the growing period? You still need to give the plant nutrient. So we give in the form of foliar. This is a bit unusual. People tak buat macam ni. But now you are in advanced physio class, you say, oh, actually you can. You can. See, it save labor, it save time, it makes you happier. Right. Okay, right. So I'll see you again at 2. Please don't be late. Kita tak nak hujan, semua orang kena basah-basah nanti. Right. Yep. 200%. 200%?
2 p.m. 100%.
Jadi kita pergi terus.
We need to settle benda tu. Ke telefon kau tak boleh pakai? Tu Google?
Kalau dia tak hujan pun, kejap lagi setengah jam lepas tu dia hujan. Like cepat.
You know what? Just be in case, just bawa payung. Okay. It's a bit unpredictable kalau macam tu. Right. It won't be long. I think we can finish around under 1 hour. Half an hour we should be finish that. Do we have extra sprayer? Then we can split nanti untuk because you need to learn how to mix the foliar fertilizer. Alright? Okay, I'll see you then. If anything, we'll just announce in the group. In the space.